welcome. My name is LaMartina and this is episode 8 of my series Neurons, in which I talk about neuroscience and neurodiversity. This episode will be about the synaptic transmission of impulses and potentials from neuron to neuron via the synaptic cleft. But first, a little bit of information about this series. This is a series in which I talk about neuroscience and neurodiversity, and more generally it will end up hopefully being the neuroscience of neurodiversity. Why brains are different between people and what actually is the neurological difference between the people. This will be mostly focused on ADHD and autism because that is what I suffer from and what I have experience with. The series contains tips and tricks for dealing with your autism and also these neuroscience videos. So now I will get in and start with a little bit of information about the human brain, which contains at least 1 billion neurons, and each of those needs a way to contact and send information to other neuronal cells, or neurons for short. It needs a way to influence those other cells, and more specifically this is done by transmitting either electrical signals or chemicals between neurons so that an action potential or a little electrical current can be sent from one neuron to another. And for the rest of the video and what I already did before, I might be looking down here, because I have a bunch of notes here, so I am sure I can say everything I want to say and it being as clear and concise as possible without me losing track, because ADHD and losing track is a thing. So I will now go on about the synapses. There are two types of synapses, electrical and chemical, and I will talk a little bit about both, but more about the chemical ones as opposed to the electrical ones. I'll show some pictures and drawings from the neuroscience book I had and used and studied from during my neuroscience classes, because I think they are very clear and they illustrate very well what I mean. So I'll start a little bit with electrical synapses, in which current flows, or potentials that flow with ions. They flow through channels from one neuron to the other, and those channels form pores between the cell membranes of the neurons, and those are called gap junctions. And like I said, they directly connect two neurons with one another, or more specifically, they connect the inside milieu of the two neurons with one another, which makes for a very fast and efficient transduction of the neurons and other stuff, because gap junctions are a way to easily move molecules and ions and stuff between cells. These specific connections between neurons are called connexons, and they can transport energy, like the energy the cells work on, like ATP, that is burned in cellular metabolism, as well as ionized molecules of calcium, potassium, and to some extent also sodium as well. I won't go into depth about how action potentials come into existence right now, because that might be a topic for a future video. So those current flows in electrical synapses are directly moved from one neuron to the other. And I already mentioned one positive effect this can have, like it can be transmitted very fast. But another consequence of this is that the flow can happen of the ions can happen in two directions, which means unless there is a blockade or some kind of way that the connections only work one way, the electrical current can be transmitted both ways, when normally a chemical synapse only works one way. And this can be useful for synchronizing groups of neurons, like the neurons in the cerebellum, or the hindbrain, I think it's called in English, but we always call it cerebellum in our studies, that regulate breathing, because that is not just one neuron, that's a whole group of neurons that controls all the muscles and the subconscious rhythm of your breathing. And because they are connected in this way with those tight gap junctions, those neurons can work together and change their current and their potential really fast together because it happens almost instantaneously when it flows. We also have chemical synapses, and I want to talk a little bit about the basics before I go into depth. Those synapses have two you can see it as two different parts of the cell that are linked together with proteins on the outside so that the synapse stays in place. There is receptors on the receiving end, which is postsynaptic, like I said before, and a way to put in neurotransmitters or chemicals that send a specific signal on the other end, the presynaptic end. At the receiving end, the chemicals are called neurotransmitters, of course, that was already obvious. They are changed into an electrical signal by opening channels that let in ions by binding to a receptor. And there are more than 100 known neurotransmitters, but their effects can be somewhat summarized in two effects. They can either be exciting or perpetuating the potential that was going on previously in the neuron, or they can have a depressing or inhibiting effect on the receiving end in which the potential doesn't go on. And there's also a rule of summation of what all these synapses bring in activation and inhibition. That makes that if the sum of all those effects of the neurotransmitter is high enough, an impulse will go on, potential will go on. But if it doesn't reach the threshold, it will not. But I will now start in a little bit more logical way with this. As you can see, the space between the two synapses in this picture I already had up on the screen. And this space is called the synaptic cleft. This is also the place where neurotransmitters are released to transduce the impulse from one neuronal cell to the other. What happens is when the potential arrives at the synapse, it will make the neurotransmitters that are stored in vesicles fuse with a membrane, and so they get released in the synaptic cleft. And in the synaptic cleft, they can bind to receptors for that specific neurotransmitter, and there's a bunch of them. I might later on go into depth about opioid receptors, because that is where the stimulant medication of ADHD has its effect on. But for now, they bind to the receptor on the receiving end of the synapse, the postsynaptic end of the synapse, where they will either directly or indirectly open and very, very sporadically close ion channels. And depending on what kind of ion comes in or out through the channel, the neuron is inhibited or excited, and an impulse will or will not appear. But not only can neurotransmitters and the effects affect other neuronal cells, synapses also end up on the different kinds of muscle tissue we have in our body. And by using specific neurotransmitters that excite, we can control muscle contraction in a skeletal muscle by choosing to move that limb. Of course, it's not a choice when it's a reflex, but you get what I'm saying? But it also, in a way, controls how our heart beats, how we breathe, and even how our intestinal system works. It controls a lot of stuff. And now, I already mentioned the vesicles that the neurotransmitters are stored in. They are temporarily used to store it until the neurotransmitter can be released. And they are made of the same membrane material as the cell membrane. So what happens when the neurotransmitter needs to be released? They fuse with the membrane. There's a whole docking and fusing mechanism for that, and I'm not going to go in depth for that because that has a lot of protein names and structures and such in it. So there's a mechanism for that. Then the neurotransmitters are released in the synaptic cleft and they bind to their specific receptors. Sometimes they have a receptor on the cell that releases it, so that a reuptake mechanism can take place again, so that the neurotransmitters are taken up by the cell that released them, so they can be used again and don't have to be reproduced. But there's also neurotransmitters that are very rapidly degraded, so that their effect doesn't last very long. And we have one type of medication I want to mention here that directly affects the reuptake mechanism of a specific neurotransmitter. It's SSRIs that are used against depression. It says selective serotonin reuptake inhibitors. So they inhibit the reuptake of serotonin in the brain, which generally makes the people who use it and who it works for, they feel more happy when they take it. Especially over the long term, it can change the way the brain acts. So that is how those medicines work. They also, of course, have a lot of side effects, but that is not what this this is about. This was about the reuptake of the neurotransmitter, and they can be degraded. I am checking now what I still like. I have mentioned everything I want to mention, and I can only tell in this video. I can tell so many more things, but I will keep that for later videos because this video is already 15 minutes long. So I will say this about the fact that the synapse needs to be held together and that it happens with proteins. Some of those proteins might play a role in how autism develops and expresses itself. I once, together with someone else, wrote kind of a research review paper on that. So this might come up in a future video as well, if you want to see, and if you don't want to see, I want to share it, so I will share it anyway. But this is all I have to say for now. Thank you for watching.